Microdroplets, tiny liquid compartments on the order of picoliters, have emerged as highly controlled reaction chambers for quantum-enhanced chemical sensing. Recent research has demonstrated that embedding quantum sensors inside microfluidic droplets enables ultra-precise measurements of chemical and environmental properties at the microscale. But how does this work, and why is it revolutionary? A key breakthrough in this field involves the use of nanodiamond particles containing fluorescent nitrogen-vacancy HEVO centers. These nanoscale quantum defects exhibit remarkable magnetic and electric field sensitivity, meaning they can detect even the smallest changes in local conditions. By encapsulating in-V-dope nanodiamonds into flowing microdroplets, researchers have created a mobile, high-throughput quantum sensing platform capable of monitoring real-time chemical reactions, biomolecular interactions, and environmental contaminants with unparalleled precision. One striking example of this approach is in chemical field detection, where the fluorescence of NV centers shifts in response to pH changes, ionic concentrations, or external magnetic fields. This means that quantum-enhanced microdroplets can function as ultra-sensitive probes for single-cell metabolomics, detecting even subtle biochemical fluctuations within living cells. Imagine a system where individual cell secretions can be analyzed at the quantum level in a continuous high-throughput manner. This could transform disease diagnostics and personalized medicine. Beyond biological sensing, this technique offers game-changing applications in portable and on-chip chemical analysis. Traditional analytical instruments often require large sample volumes and complex processing, but quantum-enhanced microfluidics allows for instantaneous in-droplet sensing of trace-level analytes. This could be particularly useful for environmental monitoring, where tiny water samples could be tested for toxins, pollutants, or heavy metals in real time. But what makes microfluidics such a perfect match for quantum sensing? First, precise droplet control enables consistent reaction conditions, eliminating noise from sample variability. Second, the high surface area to volume ratio of microdroplets enhances chemical interactions, allowing for rapid diffusion and improved sensor response times. Finally, by integrating quantum sensing techniques such as optical spin readout and magnetometry, microdroplets become self-contained nanoscale laboratories capable of detecting molecular interactions beyond classical detection limits. The implications of this research extend far beyond chemistry and biology. Could quantum-enhanced microfluidics lead to next-generation portable diagnostic devices, real-time monitoring of neurological signals, or even on-chip quantum computing elements? As quantum technologies continue to converge with microfluidics, the ability to detect, analyze, and respond to molecular-level changes with quantum precision will open entirely new possibilities in healthcare, materials science, and fundamental physics. Quantum sensing utilizes phenomena such as superposition and entanglement to achieve ultra-sensitive measurements beyond classical limits. In microfluidics, flowing microdroplets provide highly controlled environments for quantum sensors, enabling real-time analysis of chemical and biological reactions. By integrating quantum-enhanced probes, these systems achieve unparalleled detection precision for various analytes. Researchers have demonstrated that nitrogen-vacancy (NVP) centers in nanodiamonds can be embedded within microdroplets to detect changes in magnetic and electric fields with nanometer-scale resolution. This process is similar to placing ultra-sensitive microphones inside individual bubbles to detect tiny sound variations in a large ocean, where each bubble acts as a controlled reaction chamber. Nanodiamonds with NV centers serve as quantum sensors by emitting fluorescence that changes in response to external fields such as temperature, pH, or magnetism. When encapsulated in microdroplets, these sensors provide localized, high-resolution chemical and physical measurements. The quantum readout process, enabled by optically detected magnetic resonance, ODMR, allows for real-time analysis in microfluidic flows. A study showed that NV centers in picoliter-sized droplets could detect femtomolar concentrations of target molecules by measuring shifts in fluorescence intensity. This technique is like embedding tiny thermometers inside individual water droplets to track subtle temperature variations across an entire flowing stream. Quantum coherence, the ability of a quantum system to maintain phase relationships, enhances the sensitivity of microfluidic quantum sensors. By using advanced spin control techniques, researchers can prolong coherence times in NV centers, improving signal resolution. This allows for the detection of minute environmental perturbations, including single-molecule interaction. By applying microwave pulses to stabilize NV center coherence, researchers improve detection limits for biomolecular sensing, allowing real-time observation of enzyme reactions at the single-molecule level. Maintaining quantum coherence in microdroplets is like keeping a pendulum swinging perfectly in sync despite tiny disturbances, ensuring precise measurements. Quantum sensors and microdroplets enable label-free detection of chemical and biological markers, reducing interference from conventional fluorescent dyes. The microdroplet format isolates individual reactions, minimizing cross-contamination and improving assay reproducibility. Quantum-enhanced sensing provides ultra-low detection thresholds, revolutionizing fields such as metabolomics and environmental monitoring. Quantum-enhanced microfluidic sensors have been used to detect trace metal ions in water samples at concentrations lower than those achievable with traditional spectroscopy. This is like using night vision goggles to see in complete darkness. Quantum sensors amplify weak signals that would otherwise go undetected. Flowing microdroplets act as discrete self-contained reaction chambers, allowing for high-throughput sensing without sample dilution. Each droplet carries a quantum sensor, and real-time fluorescence monitoring provides continuous data acquisition. This dynamic approach enhances detection speed and efficiency compared to bulk phase quantum sensing. Quantum-enhanced microfluidic devices have demonstrated real-time pH monitoring in single-cell environments, revealing previously unobservable biochemical fluctuations. It is like using a moving conveyor belt of test tubes, each equipped with an ultra-sensitive detector to continuously analyze changing chemical properties. Advances in microfluidic integration enable compact quantum sensing platforms suitable for field applications. Miniaturized laser and microwave sources provide the necessary excitation for NV center readout, eliminating the need for bulky optical setups. These developments pave the way for portable diagnostic tools that leverage quantum-enhanced sensitivity. A prototype handheld microfluidic quantum sensor successfully detected trace amounts of pesticides in agricultural runoff, demonstrating on-site chemical analysis capabilities. This is similar to shrinking an entire laboratory into a pocket-sized device, capable of performing highly sensitive chemical tests anywhere. Quantum sensing faces challenges such as decoherence, optical readout limitations, and background noise, which microfluidics helps mitigate by providing controlled environments. Encapsulating sensors in microdroplets reduces external perturbations, improving measurement stability. Innovations in droplet actuation and sample preparation further refine quantum sensing precision. Researchers found that encapsulating NV centers in lipid-coated microdroplets significantly enhanced signal stability, reducing noise from environmental fluctuations. This approach is like placing a highly sensitive microphone inside a soundproof chamber to ensure clear audio recording despite external disturbances. Quantum microdroplet sensing could revolutionize single-cell studies, early disease detection, and environmental monitoring by offering non-invasive, ultra-sensitive analysis. Combining machine learning with quantum-enhanced data acquisition will enable automated pattern recognition for complex biochemical processes. Future advancements may integrate hybrid quantum sensors, expanding the range of detectable analytes. Quantum-enhanced droplet microfluidics has been proposed for real-time cancer biomarker detection in blood samples, potentially improving early diagnosis and treatment monitoring. This advancement is like using high-silhouette satellite imaging to detect microscopic changes in Earth's atmosphere, allowing for early prediction of weather patterns. Scaling up quantum sensing in microdroplets requires improvements in droplet generation, sensor uniformity, and automated data processing. Microfabricated optical and microwave components are being developed to enable large-scale production of quantum-enhanced microfluidic devices. Future quantum sensing platforms will likely integrate on chip photonics and nanofluidics for multiplexed analysis. A recent study demonstrated scalable microfluidic circuits embedded with quantum sensors capable of simultaneously analyzing thousands of droplet samples. This is akin to building a high-speed train network where each carriage, droplet, contains an independent sensor, allowing for rapid simultaneous analysis of vast amounts of data.